So we will start the last session of the day before we have the nice dinner. And it's a pleasure for me to announce Emily Degenhardt. And I am sure a lot of people of us are now really interested to hear about fetal exposure and the protection of pregnant women. So Emily, the floor is yours.
Good afternoon, everybody. I feel like it's a lot of pressure on me and Sophie because we are the last ones. So we have to be entertaining so people don't fall asleep and actually pay attention. I would do my best. So I would talk about radon and pregnancy and what do we know about the fetal exposure. Keep going. Here. Yeah, okay. So I would like to start introducing what is internal exposure so that it doesn't get too complicated towards the end of the presentation. And internal exposures can occur through due to inhalation of radionuclides in the air or ingestion of radionuclides in the food and drinking water, or also, for example, nuclear medicine, where we have the injection of radiopharmaceuticals for imaging or therapies, and also through wound, so absorption through the wounds. These radionuclides, when they are incorporated, they will follow their own kinetics. And these kinetics, so how they are distributed inside of the body, depends on their chemical behavior and also the biological processes that they participate in. So because different radionuclides, they concentrate in different organs, we will have an inhomogeneous distribution of the activity of the radionuclide. So if the distribution of the activity is inhomogeneous, the doses to the organs will also be inhomogeneous. So it's not possible to just turn off the radiation. The radionuclide will be decaying inside of the body and progeny will be generated inside of the body. So when we are doing assessing internal exposures, we have to take that into account. So the activity distributed in the organs, so these organs we will consider then as okay. Okay, thank you. Then they will be called radioactive sources or source regions. We cannot put, it's not really feasible to put detectors or dosimeters inside of the organs, right? So we need mathematical models to calculate the internal doses. For that, we have two mathematical models. The biokinetic models, they are related to the distribution of the radionuclide inside of the body. And the definition of those source regions, as I mentioned before. And we also have the dosimetric models, and they are related to the position of the energy emitted by the radionuclide in the self-absorption in the organ, or also the radiation deposited in the surrounding tissues. And for that, for example, we use phantoms. This is an example of a pregnancy phantom that can be applied to external dosimetry, and will be also applied in internal dosimetry. So after the intake, we use biokinetic models to calculate the time integrated activity in source organs, source regions, which allow us to calculate the number of nuclear transformations in the source regions. Then we apply nuclear data, phantoms, particle transport models to calculate the tissue absorbed doses. Finally, radiation weighting factors to calculate the tissue equivalent dose, and tissue weighting factors to calculate the effective dose. In internal dosimetry, to calculate the activity incorporated by the body, we can estimate it by the measurement of the activity of the radionuclide in the whole body, or using biosamples like urine and feces, or also using the detectors to measure specific organs. Or we can assess from the concentration of radionuclides in the air or in the water when the biokinetics is known, as we do for radon, for example. And then we calculate the effective dose, the committed effective dose, which is the accumulated dose expected to result from an intake. Because as I told you, we cannot shut down the radiation, so it will be decaying, so dose will be delivered throughout the time. And we assume that the integration time following the intake is 50 years for workers and 70 until the age of 70 years for members of the public. We usually calculate it by the intake of the radionuclide, in our case radon, and also the corresponding dose coefficient provided by the ICRP, which is the International Commission on Radiological Protection. So biokinetic models, which is the focus from my presentation, they are, as I mentioned, mathematical functions describing the distribution of an element inside of the body. The pathway of the intake plays an important role on the distribution, because if it's inhaled, then we will need to apply the human respiratory tract. But if it's ingested, we will use the human alimentary tract. So we have different organs involved in these models. And we also need to take into account the elimination pathways. That could be feces, urine, sweat, exhalation, and so on. The ICRP biokinetic models, they are usually compartmental models, and they are elemental specific, and the transport among the compartments are described using transfer coefficients. So as you can see in the picture, those boxes, they are the compartments. They are related to an organ, a tissue, part of a tissue, some fluids like blood, for example. And the arrows, they represent the transfer coefficients from one compartment to another. And these transfer coefficients, they are related to the material that is being transferred from one compartment to another. We consider first order kinetics. It's a simplification of the real processes, but they are reliable. And also we consider that these transfer coefficients, they are usually constant with time. Well, we are all somehow talking about the exposure and how we know about the increase of lung cancer risk due to prolonged exposure to radon. Why am I talking about that again? Well, for many years, the major concern was male individuals because they were minors, so mainly male individuals. But now, as the current concern is individuals working and or living in radon-prone areas, we have women, we have female individuals being exposed to radon, so we need models for this kind of subjects. We also know that preconceptional exposure to ionizing radiation, they can cause tissue reaction and possible hereditary effects. And we need to take into account also the radon progeny and their contribution to the dose. So what we know so far, based on animal studies, is that radon crosses the placenta and reaches fetal organs. As you can see, most part of the studies are older than me, but this is what we have now. This group from the United States, they carry many studies with inhalation of radon and krypton. So they're both noble gases. And when they are inhaled from air, they reach the placenta and the fetal tissues. The concentration of krypton in most part of the fetal tissues were similar to the correspondent maternal tissue. And for radon, it crosses the placenta both ways, so mother to fetus and also fetus to mother. And it can also happen with some of the progeny, for example, lead, astatine, and thallium. So the subtask 331, which I'm responsible for, in this subtask, we aim to develop a biokinetic model for radon to assess fetal uptake after radon intakes by the mother, both through inhalation and also ingestion of radon in drinking water. And for that, we are calculating those transfer coefficients using the fixed first law of passive diffusion. So passive diffusion is how gases usually diffuse in the matter. We are considering the changes in maternal tissues and also the development of the fetus. And we are using the SAM2, which is a software that allows us to model intakes prior to the conception. So the impact of intakes that mother had before getting pregnant and also throughout the pregnancy, and chronic and single intake. So in this case, we want to calculate those coefficients for the whole term of pregnancy. How we calculate that, though, we need some reference values for reference people, for reference female. And we assume that radon enters the body through inhalation. It goes to the lungs. From the lungs, it is absorbed to the blood. From the arterial blood, it reaches the organs. And then from the organs, it goes back to the blood. So we need a transfer coefficient from radon to the tissue and also from the tissue to the blood. For the transfer of radon from the tissues to the venous blood, we need the blood flow to the tissue, the volume of a tissue, and the partition coefficient. To calculate the transfer of radon from arterial blood to the tissues, we need the blood flow to the tissue and the volume of the arterial blood. This is feasible. It's easy to find these reference values. We can find in the ICRP publication 89 to calculate for the female model. The problem is for the fetus. We don't have many data. We could find sometimes data for the masses, and then we can calculate the volume of the organs. But for example, blood flows were very hard, so we needed to do some assumptions on that. And we also have some special conditions in pregnancy, because with the progress of the pregnancy, the volume and the blood flow of some tissues are changing. For example, uterus, breast, fat, and placenta. And the same happens to fetal organs that are being developed inside of the mother's womb. So the transfer coefficients in this case, they will not be constant over time as they are for the other models. As I wanted to highlight the changes in maternal organs. So why I'm highlighting fatty tissues, some fat tissues, because radon as a noble gas tends to concentrate in fatty tissues. So that would be interesting for maternal and fetal organs. For example, for blood flow rate, as you can see in the uterus, it is almost 30 times bigger towards the end of the pregnancy. So this is something that we had to consider. And we also have the changes in the fetal organs because they are growing with time. So we propose a model. Whatever is in black in the figure is what ICRP has for the adult model, and the bluish, greenish is what we added to the model. So we started from the ICRP age and sex specific biokinetic model. So in this case, female adult. We added the uterus to the maternal systemic model. And to evaluate the fetal uptake, we added the placenta. We added the venous and arterial cord blood and some fetal organs relevant for radiation protection. So thyroid, liver, bone surface, bone marrow, lungs, and brain. And we also attached the human alimentary tract model to estimate the contribution of ingested radon to the dose coefficients. Those are our results. So we have the transfer coefficients to maternal tissues. As you can see, they are changing with time, and for uterus, as expected, this is the most drastic changes. We also have different transfer coefficients for fetal tissues, and because we didn't have much data for fetal tissues, we had to do some assumptions on blood volume, because as I mentioned before, we needed the total amount of blood, the venous blood and the arterial blood, and also the blood flows. For the lungs, kidneys, brain, and other, we could calculate it using the data that we found. But for some, like thyroid, liver, bone surface, and bone marrow, we didn't find the blood flows, only the masses. So we calculated by the mass relation. It's an assumption, but it's still reliable. Well, when we apply the model, we saw that for acute exposures, up to 1.001% of the radon intake can be found in fetal tissues. It doesn't look too much, but we have a problem when we are considering chronic exposures, because due to the gradual increase in radon activity, we can have a decumulation of radon and also radon progeny in fetal tissues over time. So the interesting thing when modeling big chains of radionuclides like radon is that we cannot only consider the mother, for example, radon, but we have to consider its progeny. So lead is I forgot the name. So lead is different from radon gas, right? They have different chemical behaviors. So we cannot assume that lead will have the same behavior as radon. So either when it's inhaled together as a progeny or when it's being generated inside of the body, they will behave in different ways. So we have now a model for radon, but we still are working on the biokinetic models for the radon progeny. So for example, for lead, we know that it behaves analogously to calcium, and we have to consider that it translocates from maternal bones to fetal bones during pregnancy, and it could be important for bone marrow doses because, for example, lead 210 has a gamma emission, and it could reaches the bone marrow since it's inside of the bone. For polonium and for bismuth, based on animal studies, we know that little activity, so up to 10%, crosses the placenta. But for thallium, sorry, it should be TL, not TH. But for thallium and astatine, they tend to accumulate in thyroid. So we will look into the doses to the thyroid as well. So we are doing 6 biokinetic models for 6 different radionuclides, and they can be applied later for other purposes as well. So my final remarks, I would like to first of all address the challenges to find reference values for the mass, blood flow, and so on, but not only that, also for biokinetic data. So for radon, we could calculate, but for the progeny, sometimes it's hard to find concentration of this progeny in tissues, in fetal tissues. Of course, we rely mainly on animal studies, but they are not, we don't have many of them. Our next steps, after defining all of the biokinetic models, we will calculate the dose coefficients for different fetal tissues, for different gestational ages, taking into account the radon progeny produced inside of the body. And the specific absorbed fractions in the fetal target organs, the ones I listed, are currently being assessed by the ICRP using digital pregnancy phantoms, as I showed in my 3rd slide, I guess. And we've been in touch with them, so they will provide us the SAFs, and after combining with the biokinetic data, we can calculate then the dose coefficients. I would like, again, to highlight that all the biokinetic models that we are developing, they can be used later on individually for each element of the radon and thoron chain. So it's a huge contribution to the radiation protection community. And with that, I would like to thank you for your attention.
So thank you, Emily. So we will start the discussion. Are there some questions from the audience here? Online? Yeah.
Questions in chat.
Okay.
In biokinetic model, what about the brain? Is there no effect of radon on the brain from blood capillaries?
This is a really good question. I mean, in the fetal model, we are considering the brain in the fetal systemic model. ICRP in their adult model, they didn't implement, but we did it to the fetal. So we will consider it as a source region.
Is there any other question? I would have a question for that. So what organ do you consider mostly at risk in the fetus?
I can show you for, personally, I'm very interested in the bone marrow because it's a fatty tissue. But as you can see for this graph, liver is accumulating a lot of radon. Yeah.
Thank you.
Thank you very much, Emily.